So I request, Dr. Uday Narayan Bhatt onto the dais. Dr. Uday Narayan Bhatt. Dr. Madhavi. Dr. Uday Narayan Bhatt is from Kopat Biological Systems. Dr. Madhavi from State Agricultural University, PJTSU. Please come onto the dais, ma'am. And Dr. B. Venkateshwara Garu, ex-director, Krida. And Professor Apparao Garu, the former Vice Chancellor of University of Hyderabad. Dr. Vimala Prakash from IPL. So this session mostly deals with biofertilizers. This is session 2 after the inaugural session. And here the speakers will be talking on the biofertilizers, its technology and business landscape. So we have almost excellent balance here. 2 speakers are from industry and 3 are from academia. So, sirs, it's one more request, again another request to complete in 10 minutes so that at least we'll give time for Q&A. So let us start with mycorrhiza and endophytes and emerging global markets, and that will be delivered by Dr. Uday Narayana Bhatt, Kopat. Sir, please.
Good afternoon to the audience and also the organizers. At the outset, I would like to thank the organizers for giving me this topic to speak on, and very good gathering about the bioproduct, the entrepreneurs here. Welcome. Next slide. Yeah, the topic is mycorrhiza and endophytes, emerging global market. And I would like to finish it in a short time because of time constraints. Next slide. Yeah, to start with, I just want you to know that, Sadhguru started his journey for creating awareness on soil health. This one, no? And his first destination was Koppert in the Netherlands in March. After London, he started it in London. And we were taking him through our experience center where he has seen the tree of life, all the microorganisms and how it's all done. And he was very impressed by what he has seen there. And we are also a part of the solution for saving soil health. So that's what is reflected here. And I think all the companies here will agree to that, because without soil health, there is no sustainability or there is no natural pollution control which is happening. Thank you. Next slide. Key market trends. In this, I would like to touch upon the mycorrhizae in particular. What we see is the phosphorus content in the soil is decreasing, and over the last couple of years, you see from 2018 onwards, there has been a decrease in consumption of phosphorus fertilizers. It's basically because of higher price rise also, and there has been about 50% of the soils in India are phosphorus deficient. And mycorrhizal biofertilizer is an answer to this particular problem, because it helps in absorption of both mobile and immobile nutrients, mobile like phosphorus, zinc, and copper, and the other immobile phosphorus, zinc, and copper, and mobile nutrients like sulfur, calcium, potassium, iron, manganese, and nitrogen. Asia-Pacific region is the highest consumer of mycorrhizal fertilizer, because Asia-Pacific region is also producing highest quantity of rice, because in paddy fields, most of this mycorrhizal biofertilizers is used. However, the mycorrhiza plays a major role because of the depletion of phosphorus and also the price increase. Next slide. Yeah. So global market scenario of mycorrhiza. There are three, high growth, medium, and low growth. What we see is this based on data from Mordor Intelligence, that 2017 to 2027, Europe is the fastest growing market for this particular products, mycorrhiza based. And however, largest is Asia Pacific. The CAGR is about 14.8%. And what we see is in 2021, the global sales turnover of this particular product was USD 271 million, which is projected to increase to 632 million by 2026. So there is a lot of scope for this particular stream of biofertilizers. Soil care is a niche thing, and in this particular thing, the mycorrhiza and PGPR has got tremendous opportunity for growth. You will see that in the forest soil, the mycorrhiza content is more than about 12%. That's why you can see luxuriant growth, even though there is no artificial or synthetic fertilizer applied. While in domesticated soil, where we keep cultivating crop over year after year, the mycorrhizal content is less than 5%. Because of that, we need to add more of synthetic fertilizers. If we had the mycorrhizal level increased even to 5%, then our application of fertilizers can come down. Why has that happened? That has happened because of excessive use of herbicides, pesticides, fertilizers, that's synthetic fertilizers. And we need to now increase the quantity of the rhizosphere with the mycorrhiza, and that will definitely make a big difference in cereal crops, pulses, sugar, and all other vegetable crops. This is basically a brief information of mycorrhiza. Myco is fungus and rhiza is roots. Mycorrhiza is associated with the roots. It has symbiotic relationship with the roots. It helps in uptake of nutrients from deeper layer, uptake of moisture, water content from deeper layers. It helps in building the plant's resistance also to certain pests and diseases. Because of its activities in the crop. So that's why, as rightly said, we need to also consider these microbial based things under the category of biostimulant also, because it has got biostimulation activities. And most of the crops, generally they are about 90% of the crops have mycorrhiza, but because of domestication, the percentage has gone down. So we, in order to recolonize them, we need to get them back from the native or, from the plants in the environment. And then there are two major types, ecto and endomycorrhiza. Ectomycorrhiza are not invaginating into the cell wall, while endo is invaginating into the cell wall. There are other types also, arbitoid, ericoid, monotetroid, and orchid mycorrhiza. So what are the benefits of colonization with mycorrhiza? In case of non-colonized thing, you would see that the application of fertilizers, they come into the soil solution and basically they are absorbed by the plants. But in case of arbuscular mycorrhizal colonization, what you see is it helps in uptake of absorption of nutrients, because these roots have got this mycelia has got symbiotic relationship with the roots, and they go to the deeper layer and then they make the phosphorus available to the crop. Similarly, they make other nutrients also available to the crop, and then also they help in biotic, abiotic stress tolerance, and this will help in increasing the availability of nutrients and reducing the fertilizer application, minimizing the pest and disease incidence because of the plant mycorrhiza interaction. Next slide. Yeah. One more topic is endophytes. Endophytes is these microbes which are in the plant system. Basically, they gain entry through seeds, through leaves, through roots, and also through stem. And this also has a similar function as mycorrhiza. Mycorrhiza is only the roots. This thing helps in nutrient uptake and defends the plants from pathogens and insects, modulates plant development, enhances biotic and abiotic stress, suppresses weed growth. So this is also one of the reasons why we should consider these things also as biostimulants. Examples of endophytes are Pseudomonas, Rhizobium, Bacillus, and there are certain other fungi also, Serendipita, Fusarium, Pinnifernospora. Next slide. So this is again domestication and long-term cultivation. Loss of endophytes has happened. Chemical seed treatment, acid seed treatment, massive use of chemicals and fertilizers. And in order to increase the endophytes, we need to take them from the wild relatives crops and reintroduce them. That is the recolonization which has to be done for the benefit of the soil. Next slide. So Coppert has developed a unique mycorrhiza locally from the local strain, and this has been done by root organ culture aseptically, and it's called as Rhizophagus irregularis, and this is deposited in Belgium depository with strain number, accession number also. And we are fully compliant with the Biodiversity Act, and because we are paying royalty also to the Telangana State Biodiversity for access benefit sharing. The R&D and QA is backed by Coppert.
Please wrap up, please.
Sorry?
Wrap up, please.
Yes, please. So this is again the details. Anyone who wants to approach us for the mycorrhizal requirement, they can definitely contact us. We have this in 50,000 spores per ml and also 10,000 spores per gram as per FCO. We have a production capacity of 10 billion spores, which can be increased to 200 billion is possible. Next slide. Yeah, these are some of the trial results. Wherein we have seen excellent results with this mycorrhiza strain. Next slide. Yeah, this you will appreciate. You can see that in the control, the root growth is about 25 grams, while in case of mycorrhiza treated plant, the maize crop is about 47 grams. You could see what opportunity is there, because all the soil, in the soil you will see most of the fertilizer is locked. This makes it available to the crop. So tremendous opportunity to reduce the fertilizer, synthetic fertilizer. Next slide. You finished. Yeah, this is the product. You should apply about 100 grams per acre. So I think if there are any questions, you can always discuss with me. I am here for 2 days, and then have a good meeting. Thank you.
So good afternoon to all of you, and I am very thankful to our university and BIPA for giving this opportunity to speak few words about the microbes for macro and micronutrient management. So mainly the microbes, they are involved in two aspects, that is fixation and mobilization. The microbes' role in soil is only on these two aspects, that is fixation and solubilization or mobilization. That is, they are mainly involved in nitrogen fixation and uptake of major nutrients, and nutrient mobilization and promotion of shoot and root growth, disease control or suppression. This is the main role of microbes to increase the availability of macro or micronutrients in the soil. Next one, if you take the nitrogen in the major nutrients, so the nitrogen is mainly, it is fixed by the microbes, and that fixation is mainly by symbiotic fixation in pulses, whereas asymbiotic in other commercial crops and associated symbiotic in cereals and millets. Rhizobium and bradyrhizobium, these microbes, they are involved for symbiotic fixation, and Acinetobacter, Baserincea, they are involved in asymbiotic fixation. Symbiotic means there is a mutual benefit for those two parts that are involved in the utilization. So in the pulses, the rhizobium, it produces nodules, and it will fix the atmospheric nitrogen. That nitrogen, it is utilized by the plant. In the other way, the plant, it supplies the carbohydrates to the microorganisms. That is the symbiotic association. If you take any nutrient mobilization or for transformation, if you see from the atmospheric nitrogen, the nitrogen, it is fixed and it becomes decomposed, and it is finally converted to ammonical and nitrite and nitrates by involvement of nitrosomonas and nitrobacter, that is the bacteria involved for final conversion of nitrogen into absorbed form of the nitrate by the plants. So this is in this the denaturifying bacteria involved for the losses and the beneficial organisms, that is nitrifying bacteria involved for the availability of nutrients, that is the nitrogen. Okay. Next, coming to the phosphorus mobilization. Phosphorus mobilization, it takes place by different processes by phosphorus mobilizing bacteria. So these phosphorus mobilizing bacteria, they will produce acids, carbonic acid and hydrogen sulfide, and these are all produced by respiration or assimilation process, and some of them, they will act as chelates also to solubilize the immobilized phosphorus that is present in the soil. So because of all these process, the protonation occurs in the soil. So because of this protonation means there is acidification or release of hydrogen ion. So in this process of release of hydrogen ion, there will be the development of acidity. Because of development of acidic nature, the mobilization or solubilization of fixed phosphorus that took place in the soil. That is the phosphorus solubilization by involvement of phosphorus solubilizing bacteria, that is Pseudomonas or Bacillus or Rhizobium or Penicillium or Aspergillus. These are all involved for mobilization of phosphorus to make the release of acids and to act as chelates and also to act as to release the hydrogen as a proton, which will create the rhizosphere atmosphere in the acidic nature, or that acidic conditions, it will solubilize the insoluble phosphorus that is present in the soil to soluble one. So that these are the phosphorus solubilizing bacteria which will give the availability of phosphorus to the plants. If you see the mycorrhiza here, the VAM, already it is explained by previous speaker, that is vascular asbescular mycorrhiza. It will interconnect with the external hyphae and it is mainly involved in P absorption and the availability. So those are the pictures where the treatment of mycorrhizal treatment. With mycorrhizal treatment, there will be the more growth of roots when compared to the non-microbial. Here also there is the development of root. If you take the plants also, there will be the improvement with phosphorus solubilizing bacteria when compared to non-solubilizing bacteria. So this is the mechanism how the phosphorus, it will be mobilized with the microbes in the soil. Next, coming to the potassium. The potassium solubilization bacteria, it is mainly are Bacillus species. So only 1 to 2% of element is available in the soil and remaining will be fixed. So the mechanism how the phosphorus, it will be solubilized by the microbes is by involvement of production of inorganic and organic acids and acidolysis, polysaccharide production, chelation, complexolysis and exchange reactions. These are all processes are involved to convert insoluble form of potassium into soluble form of potassium by involvement of microbes. Because of these processes, there will be the availability of insoluble phosphorus to soluble phosphorus by involvement of these microorganisms, that is Bacillus species. So the potassium solubilizing bacteria are effective in releasing potassium from inorganic and insoluble forms of total soil K through solubilization. That is main example is Bacillus circulans. This is how the potassium, it will be mobilized in the soil by involvement of the microorganisms. Next one is in the micronutrients, the main one are zinc and iron. The zinc mobilization, it will take place so many species of bacteria, that is Bacillus, Pseudomonas and Serratia are main. So what they will do is, they will lowering the soil pH by producing acids like gluconic and acetic acid. These gluconic and acetic acids are main acids which will solubilize the insoluble zinc in the soil to soluble form and make it available to the plant. Because the zinc use efficiency is very less, when we apply the zinc to the soil, its efficiency is only 1 to 2%. So the availability or use efficiency of zinc, it will be improved by application of the zinc solubilizing bacteria like Bacillus, Pseudomonas, Serratia. So they will release the acids like gluconic and acetic acid.
minutes.
Next one is iron mobilization. The iron mobilization in this, the bacteria or the species involved are Rhizobium, Bradyrhizobium, Pseudomonas and Bacillus. In this, they will mainly produce the siderophores. Siderophores is low molecular mass proteins. These have high affinity to act as chelates. So these chelates solubilize the insoluble iron in the soil to soluble iron. So the iron solubilizing bacteria are mainly solubilize the insoluble iron in the soil to soluble iron by producing the siderophores. So this is the process how it occurs by involvement of those ones. So these are some of our experimental results where there is improvement in the yield by application of 25% of biofertilizers when compared to the 100% RDF. So there will be the improvement or on-par yields with application of 25% biofertilizer and 75% recommended dose of fertilizers. Thank you.
This is the topic given to me. In fact, as you all know that dryland agriculture is a very important part of our country, and this statistics would tell you, almost 50% of the cultivated area is dryland, and it produces 40% of the food grains. And these are the important crops that are grown and important organisms that are relevant. And I would like to take back a little bit, to you, to the actually when the research work started. It was started as far as back 1950, when the All India project started in India on different crops. So that is how the strains, recommendations, multi-locational testing on crops, all that has been happening. And initially there was little bit, the results were not very encouraging. And the farmers were not really forthcoming to take this technology because of the inconsistent results across the locations, across the crops, etc. And also at that time, there was very poor linkage between the industry. Industry was also very limited. In fact, I remember when I was working in the 80s, there was only, most of the industry was in Maharashtra or Tamil Nadu. There was hardly any industry in the rest of the country. There was very little awareness on this. But after that, after 2000, this is the second phase I would say, that the phosphorus solubilizing bacteria has been performing well. Mainly because most of our soils, there is a lot of fixation of phosphorus because we are adding a lot of P after the green revolution. As a result, we see some good results with phosphorus solubilizing bacteria, then later on potash, zinc, etc. And then PGPR's era came. And that is how the interest on biofertilizers got revived, especially in case of drylands, And also the private industry came in and then they have improved the supply chain a little bit, and then the situation improved. And the yield benefits and economics in the early phase, again, were not very encouraging, that the benefits were mostly about, in most of the trials, if you see, I summarized, about 10 to 15%, yield increase, and that too not in every location. And many times nowadays our farmers' attitude is that unless the substantial benefit is there, they will not try to venture into a new product, except the seed which is very easily adopted. And also in drylands, the biggest problem is, it's not the culture alone, it is also the moisture that is very important. And I'll show you one slide where if you inoculate and along with that you follow conservation practice, then you will get the much better benefit of yield improvement rather than inoculation alone. And then as I said, in the current phase, especially the zinc and phosphorus, we are now promoting where there is a deficiency, and as a result, the Bacillus Pseudomonas era, where a lot of, hormonal and other benefits have come. And now consortia again is another innovation that has been coming up. And now in the last, say, about 5-6 years, people have done the resurvey again. Many essays, earlier they were saying is only about 10% adoption, But now there is a, results or data coming up. Now the adoption has improved up to 25%. Of course, in agriculture, 25% is not a small adoption. No technology can be adopted by 100%. There will always. So this is, of course, a random survey, maybe not applicable to all products, but that is general, kind of feedback. Now, this is just to show you some of the early results in 90s, In 90s, the work was done. In fact, I would like to acknowledge the public sector contribution in research and biofertilizers. Many times, we don't acknowledge, lot of work was done in 80s, 90s, and at that time, all India level trials were conducted on different rhizobia, PSB, etc. And these are all the pulse growing areas, and the average improvement you can see areas as low as 8% to as high as 24%. Similarly, in our own dryland CRIDA network, in 95 to 2000, lot of trials were done on various phosphorus solubilizing bacteria, and you will see some locations non-significant, but where are other locations 10, 15, 20% benefits were found. And this is the slide I was talking about, that if you follow soil conservation and water conservation practice, then you will get much better results as compared to, just normal flat sowing. Flat sowing, contour planting, sowing flat, and conservation for rows. So inoculation in dryland agriculture, inoculation alone will not do. Along with that, you have to also integrate with a good agronomic practice. How actually a good agronomic practice, there are many things, but most critical is the soil and water conservation practices. And in 2011, we started work on climate change, whether microbes can have any role in amelioration of the climate change impacts. And this was our first experiment done by one of my students, that we wanted to see whether microbes can alleviate plants from heat stress. And this is a small experiment which gave us clue to us that we grew sorghum seedlings under high temperature, elevated temperatures in a controlled conditions. And we found very surprisingly that when you inoculate with Pseudomonas, which was again isolated from a very high temperature tolerant, high soil in Rajasthan, and the plants were able to survive almost 7 days more than normal control. Otherwise, they also ultimately died. But 7 days more than the normal plants which were not inoculated. And we also tried this with drought also, just withholding water. And there also we found that you can survive plants more than, of course, 1 week to 10 days more than. So this gave us a clue that, organisms not only provide nutrients, some of the organisms like Pseudomonas and Bacillus, but also they protect plants from heat stress and drought stress. And this year, for example, we lost a lot of wheat in North India due to heat stress. And now we have these cultures in CRIDA, anybody interested. And I would advise that, these cultures can be formulated either as a seed coat or as a foliar spray, and then see whether heat stress can really be controlled in the field condition. In fact, my student did some work on how actually this heat stress protection or drought stress protection actually happening. Two mechanisms we found. One is the microbes are inducing plants to produce heat shock proteins, which were otherwise not being produced. Second is, these are producing very amount of extracellular polysaccharides, which are aggregating the soil particles in the rhizosphere. So when the soil particles aggregation is more, then they store more moisture, in the soil. So that is how, it's a multiple mechanisms through which actually these organisms help plants to cope with stress. Morning, Dr. Malhotra said, we must work about the mechanisms. In fact, this kind of work we did almost a decade ago, and now, of course, some students are still continuing. But there are a lot of ways in which plants help in abiotic stress. Dr. Malhotra, I would like to also draw your attention that, we have biofertilizers, we have biostimulants, and we have bacteria, fungi for plant protection. But this is a new way of protecting plants from abiotic stresses, drought and high temperature, which is very important nowadays with climate change. Now, in what category you will put them is something we have to, deliberate, whether they are biostimulants or biofertilizers, because these are non-nutritional benefits or effects on the plants, which are very critical nowadays with climate change. Every year we have either waterlogging, drought or floods. And these are some of the organisms when we tried in the field conditions also, they did very well under, without irrigation. we also did in field conditions under completely rain-fed conditions, and this is the kind of effect you will see. And in the way forward, what we, this is more of a slide with a business orientation. What I would like to say is that we must capitalize on to promote our biofertilizers on the government's interest on reduction of fertilizer subsidy, and also, the whole global gap, India gap, etc. And also we must perhaps demand a separate set of budget allocation for biofertilizers, because when the government is giving so much subsidy on chemical fertilizers, even 0.1% of that, if you can allocate to biofertilizers, their promotion, their R&D, etc., I think it will go a long way. This is what I think BIPA should, argue for. Then the natural farming people are totally against including biofertilizers. I think that mindset we have to slowly change. And then multiple organisms, especially, I would like to say some of the crops like direct seeded rice, aerobic rice, export-oriented vegetables, spices, etc., where the residue is a problem, there we have not really explored, how to use biofertilizers and whether we can avoid these problems which are mentioned. And then in acid soils, we must also try better methods of, seed coating, like pelleting with the lime, etc., so that, the organisms will not. And I would like to give a very, very solid recommendation that instead of promoting KSB, ZSB, everything, everywhere, we must make use of the Government of India ICR prepared maps. ICR has prepared, taluka level maps on nutrient deficiencies, both potash, zinc, sulfur, etc. And we must place our product according to the agroclimatic zone and deficiency maps. Then we will be able to target the right place and the right crop and right farmer. That way we will be able to, generate more, not only business, but also will serve the country. Then for perennial fruit crops where inoculation is not very possible, we must try some foliar products, I think that is where we are. And then for fruit and forest, nursery is a big business in our country. Nursery is now very big seedling production. Now we don't have very good products for seedling production. And then another very important recommendation, Dr. Reddy, I would like to make is, our culture collections in the public system are very poorly maintained right now. People don't bother to subculture, they don't maintain properly. So you must ask in terms of infrastructure investment from the government side, which no private company can do, to establish the world of the state-of-the-art best collection in the world, so that all the cultures are maintained for prosperity. I think this is one of the most important things you must argue and get it done from the government. And Mr. Agarwal, this is a slide for you. You said that, every speaker must link your speak with the theme of the conference. So this is what I did. Biofertilizers in India has a long history. I would say next to US, India is the only country. In 1920, when indigo crop was grown in India for dye purpose, at that time, farmers in Bihar crushed the nodules and made a suspension and used inoculated. So it's very, very, well known and historical. And then we have a very good SAU setup. We have very good KVK setup.
Sir,
and
please
we have
conclude,
very good,
sir.
Yes. So we have very good R&D setup. And so therefore, taking all these advantages, I would say this is the right time for India to push these products and become a global leader in bioproducts. Thank you very much.
Thank you, sir. Thank you so much. Invite Professor Dr. Apparao Garu, the former Vice Chancellor of University of Hyderabad, to deliver a talk on nano applications in agriculture.
Thank you, Professor Reddy and Dr. Kerka Reddy, co-panelists and members. I'm happy to share a few things about what we have been working on the nano Agri-Inputs, maybe for the past 10 to 12 years. I'm not going to share the results, don't worry about it, not about research per se, I'm not going to talk. I have planned to brief about how this nanotechnology has started some 50 years ago, and from this nanotechnology, at which point in time, I think the slide is, there is a lot of slide on this side. It started in 1970s, And the little of activity we can see, yeah, good, thank you. We can see Agri-Inputs have started sometime around 2005. And around this time, the Department of Biotechnology has started a task force on nanotechnology. And in 2010-11, somewhere they have sanctioned as a major research project. And from there, we started working in it. And there is a lot of information about the enhanced nutrient uptake, and then about improved growth of roots and increased germination rates. These are the things that were shown around 2005 using these nano products. And as we passed to 2010, we can see uptake into the roots, taking minute, even among the nanoparticles also using the quantum dots, the minutest ones, ranging between 2 nanometers to 10 nanometers range. The uptake from the roots to the shoot have been demonstrated and promoted the safe crop production. And then finally, we also have some of these things we have made contributions. Enhanced disease resistance has been shown, making use of the nanoparticles. This is the journey. And as we can see, the applications of the nanotechnology in agriculture on both in crop breeding, which we are not talking about, biotechnology, crop breeding, we are not talking. It is getting more and more towards this 2020. It is, the applications are increasing and more information is coming out on that. And major areas where we see is they are seen for the enhanced plant growth. They are also seen for germination. And the 3 different kinds of things that we broadly can see are the nanopesticides that and nanonutrients and nanocarriers. These are the areas where major focus is there these days. And they are also helpful in soil health management. Just a little of understanding for all of us. When we have micrometer kind of particles, the number of particles that can be sprayed onto the leaf is limited. And when you have the smallest particles, the number of particles that you can spray on the leaf are more. And because of that, there are some advantages. That is, what is the actual advantage with the nanotechnology, where we have increased permeability, improved coverage of the surface, and then you also have improved absorption. These are the 3 major advantages that we will have. And these are applied making use of different types of nanomaterials, as in the right and left-hand center, you can see different types of nanomaterials in different forms, either in a particle form, emulsion form, layered systems, or porous-based, or capsule forms, or fibers. These are the different kinds of formulations that people have developed and used as agri-inputs. And for all the kinds of applications that are shown or centered around, you have chemical pesticides, the pheromones inserted in that, fertilizers inserted in the biological pesticides into that, all that has been happening. And these are what the advantage of this is. There are other things that I will discuss about the concerns that we have. Reduce the degradation caused by the environmental influences is one saving that we can have, and achieve the target controlled release of the particles by these nanomaterials is another advantage that we have, which we have. The time is very limited. I'm ready to discuss with anybody. We have some important information around all these things with our research. This is the concerns that I'm talking about. The high reactivity and variability sometimes we consider as an advantage, but it is also is a matter of concern. Environmental impact of these is being assessed. There is some positive information, but there are also some concerns around that environmental impact, safety concerns for the workers who are using that, and also the safety concerns for the consumers. All these are major concerns that are surrounding it. But on the left-hand side, when we see the green ones, we are always attracted towards making use of these technologies. And these are the ways in which the fertilizers are being used, and there are advantages. Leaching losses can be minimized, and there is protection for the groundwater, and you have high utilization rate of the fertilizers because of the retention when we use these nanomaterials. This is more clearly shown in this particular slide. What you can see, the retention, because of the usage of these nano formulations of that, there is a retention of that. This is actually it is around this area we are working. That's where I thought I'll spend the 10 minutes that he has given. I have taken 4 minutes there, and I will take 3 minutes for what we are doing, and so that I can finish it off within 10 minutes. We are working with a natural renewable resource like chitin, which is taken from crab shells. And now I will show in the next couple of slides, you have more clear picture of this upscaling of the chitosan nano fertilizer, which has been already planned and executed by people. And there are several good advantages of using these kind of materials for the slow and controlled release or the targeted delivery of the nutrients. And this is primarily the process by which the chemical, the chitin is extracted from these natural resources, and a de-acetylation of this chitin can help us to develop chitosan. Chitin and chitosan are very close, and only the difference is de-acetylation process. This is the process that I'm talking about. From about 100 tons of this renewable source, the waste that is coming from the shrimp industry, you can get about 2 to 3 tons of this chitin, or you can also convert into chitosan, and that mixed with these active ingredients using another kind of a reactor, and then after proper washing of the processes, and then centrifugation and lyophilization, do all the quality checks, and then make use of it as a nano fertilizer with active ingredients associated with the chitosan or chitin, whatever we would like to make use of. I would like to add, as a professor, we always would like to talk about something what is coming recent, and you can see all that is not that you see only visually outside. There are a lot of things that are happening inside the plant when we are applying these materials in a nano formulation. The best of the things for every such benefit that people have seen, lots of the genes that are responsible and the genes that are overexpressed in response to this nano Agri-Inputs treatment has been demonstrated. That is a clear evidence that there is a molecular change that is happening inside the plant, and because of that, we are seeing all these benefits outside the plant, And this is another latest discovery that I would say all of us should keep in mind, that it is when there is light sufficient condition, it is possible by making use of these the down conversion nanoparticles and up conversion nanoparticles, 2 different types of nanoparticles, making use of that. Under light insufficient conditions also, we will be able to enhance the photosynthetic rate in the plant. That is the beauty of using these nano inputs. Yes, I am done. Futuristic approach for a targeted delivery. This is something which I was telling you, that we have some interesting things to share. The chitosan-based nanoparticles that we have used directly got targeted into the chloroplast when we have done a lot of experiments. I'm not showing on any of those results here. The reason is that the material that we have used inside, that is the herpin molecule, targets this into the chloroplast. But here, the model that is shown, you can either use a mitochondria recognizing motif or a chloroplast recognizing motif, and you can make sure that your nanomaterials can directly enter into the organelles of the plant cells. So our lab experience, I was telling you the last 10, 12 years, whatever we have been doing very quickly. This is the process that we are talking, herpin loaded. Herpin is a bacterial protein which we have loaded into chitosan, and chitosan nanoparticles, that is a DVT project which we have completed, on which we have filed 2 patents on that, and we also have published that information. It is in the public domain. Green synthesis of selected nanofertilizers, which is an ongoing project. And somebody, as I said, I'm not talking about the results here. And the scale-up of the field testing of the nano Agri-Inputs is the 3rd major project that we are about to start. Actually, it is just started, and it is, interestingly, jointly with our chairman here for this session, Dr. K.R.K. Reddy, the MD of this Sri Bioaesthetics, is our industry partner, and he is also supporting the grant apart from what the government is giving. And we have another partner, the ARCI, who are also experts in manufacturing of the nanomaterials. With this, I would like to thank the BIPA, the organizers, and all of you for the time that you have given me. Thank you very much.
Thank you, sir. Thank you so much.
being given the toughest job of talking about biofertilizer regulatory aspects. We are not having the presentation, it's okay. I'll just continue. And the first thing is that I thank Dr. S.K. Malhotra sir and Director NCOF for covering most of the points. What I'm going to talk is the industry pain point. The existing FCO specification of the biofertilizers and what are all the anomalies, the gaps that is there in the presentation, in the FCO specification. Sorry, the presentation is not there. I'll just, there are, in case of biofertilizer specifications, we have got almost 11 specifications is there for biofertilizers. But compared to the chemical, you have got rhizobium.
Dr. Vamla, is it?
Not this one, sir.
Okay.
I sent to, this is for the corporate presentation.
Corporate, okay.
It's okay. लैपटॉप में है लैपटॉप उनके पास है ना शैलेंद्र कर दूंगी इसे So there are around 11 FCO specifications are there. In that many of the points are not covered. So there are certain reasons for the product failures. When you, it is not because of the quality, but due to other parameters also there is a reasons for product failures. One is that inappropriate parameters that is mentioned in the FCO specifications. Some protocols are outdated. There is only single protocol to test all the microbial formulations. So that these are the pain points what the industry is facing now. Second is that the testing lab. See, we make a very good quality product, we send it. But when the testing lab is not well equipped, the product gets failed. Second is that the technical people who are testing our products, sometimes they are not able to meet the product testing parameters. That is also another reason why there is a failure of the product. So what we want to say, this is an industry pain point. So we want the industry and the association to represent it. What is the way forward? How to fix this gap? So coming is that there are certain protocols. For example, in case of pipettes, it's only mentioned as you should have 1 ml and 10 ml pipette, but the volume you have to take is 0.1 ml. So that is another thing. Certain parameters are very outdated. For example, rhizobium seed treatment, you have to...
Dr. Vimala is there.
Yeah, great. Thank you. So just go next. Next.
Next, please.
Next.
I think you can use that.
Yeah, then next. It's not. Yeah. Previous slide, previous slide. See, there are certain anomalies and gaps in the protocols that as an industry and the association, we have to take it forward. One is that the product testing protocols, the parameters, what is there in the specifications. Third is the specification itself and the protocol itself. Then, for example, in case of biofertilizer, I think you have told that labs are there, but many of the labs are not. This is the industry pain point, I want to take it. The labs has to be exclusive labs we require for biofertilizer. Because some of the quality products, when we send it and it is not clear, then the entire sale and the industry and the business, everything suffers. So we need a very good exclusive testing lab for biofertilizer. Coming to the second is that I told you there is one protocol that is mentioned for one microorganism, for one nutrition. Suppose my PSB, it requires another incubation temperature. For some other industry, the PSB what they are using may require another incubation temperature. But I have to follow the same protocol across the industry and across the product and across the nutrition. That will be very difficult. So what we are requesting is that one of the pain point is that if we have a very good DSCR recognized lab, why don't you use our protocols? You can, because this is a protocol that we have standardized for our products. So please give us the opportunity to represent this so that we can use our protocol. That is one thing. Next. That's a suggestion. Yeah, coming to the biostimulant, these are the present 8 classifications. In that there is cell-free microbial products. The metabolite which the microbe is secreting, that has been included, but microbe has been excluded. As Dr. S.K. Malhotra sir has covered, there are n number of microbes. It's a natural, nature to nature is the concept. N number of microbes are present which can act as biostimulant. They can be never be a biopesticide or grouped under biofertilizer. But we are completely... चल नहीं रहा है. We have completely ignored these microbes, so definitely the microbe should be a part of a biostimulant. That classification is very much required because in FCO there are only 11 groups, and in case of biopesticides you take only 35 microbes, but you see the complete microbial population that is present in the nature. There are so many beautiful microbes are present which can act as biostimulant, but we are not able to get into that. I think it's something is going to happen, means it's very good for the industry. Next. Yeah. With respect to FCO also, we have got very limited representation, that nitrogen fixation, PSB, potash, zinc, that's all. We do not have any classification or specifications for iron and sulfur, the secondary one like calcium, magnesium. So these are microbes, we have microbes, and these microbes have to be included under FCO or any other specification, because we come out with a very good product which is able to provide iron and sulfur nutrition, but there is no classification in FCO. We are out of sales. We are finding difficulty in getting a sales permission. That product is not able to come to the market. So these are the anomalies which has to be taken care of. That is a pain point for the industry. Next. Yeah. See, microbes, yes, microbes is something biological is picking up.
2 minutes.
You have to give me
It's
more.
given considered.
Okay. See, microbes are picking up, biological market has opened up, but yet microbes can be a standalone. Yes, they can reduce the usage of chemical fertilizer by 25% or 30%, but their efficiency definitely increases when it is mixed with an elemental form. Now, we have done some trials, we find, yes, there is good amount of nutrient uptake efficiency is there when you are mixing a zinc solubilizing bacteria with an elemental zinc. When it is getting chelated, this will act on it. But government or any clarity is not there, so we would like the industry to represent it or the association to represent it to have a clarity on it. Next. Another thing is that biostimulants, now we have got 8 classifications like humic acid and all those things, humic acid, botanical extracts, but you mix microbes with these biostimulants, definitely the efficiency of the biostimulant also will increase. That is also, there is a gap, that is one thing. Just one point I want to add it, sir, because my presentation was not there, just only one. In case of FCO specification for liquid biofertilizer, we have got a specification for pH. For example, it should be from 5 to 7.5, 6.5 to 7. But that pH for liquid for biofertilizer has to be little bit looked into. The reason is that, see, I have an azospirillum, I am able to formulate it. I have a shelf life for 1.5 years or 2 years, but the pH is not coming in that range. 6.5 to 7.5, I have to drop that formulating agent or the formulations, which I am using it to make the organism to survive for a long time. So that has to be taken care. Really, because that's a very good product. I have a shelf life, but if I send it to the market, because of this pH range, the product may fail. The labs are just, because all the government, they just stick on to the rule book. So many of the time, this is the industry pain point what we are facing. And sorry for the mishap in the beginning. Thank you for the opportunity.
Great. Thank you, ma'am. Thank you. So my topic will be on microbiome engineering, the trends and prospects. So may I have the next slide, please? Thank you. Yeah. See, nutshell, we are all, I think the scientists who spoke about the microbes, their role in nutrients fixation. Maybe the next speakers will be talking on the biofertilizers and biopesticides and biostimulants. All about, most of them are microbials, or maybe some organic biostimulants. So when we talk about these microbes, so these microbes, we know since long, microbes can fix nitrogen or maybe the phosphate solubilization, some mobilization. So like that, now the concept is from microbe to microbiome. That means it is a holistic approach where you will be using a set of microorganisms. That means it's a consortium approach. That means each organism may be having one or many functions. So likewise, we can have an organism or multiple organisms having the multiple functions. What are those functions? Those functions are beneficial to the plant. So therefore, the definition is a set of microbes are associated with in a habitat or maybe a plant or animal insect. If it is a plant, we call it a plant microbiome or insect microbiome or human microbiome. So in nutshell, the definition is all kinds of microbes associated with a particular habit or habitat or a organism. So that is called as microbiome. So how these microbiomes can be exploited here? So in our business, most of them are all from the business community here, the philosophy is here, all plants, wherever they grow, it may be a desert ecosystem, cropland ecosystem or marine ecosystem, every plant, it's not only plant or animal or insect are associated with the microbes. There is an excellent connectivity or actions and interactions between these microbes. So that is our research philosophy or business philosophy, that therefore these microbes are ubiquitous and without microbes the survival is very, very difficult. And these are the microbiomes I have already mentioned. These are the microbes which are associated with a plant or maybe a part of the plant. Maybe it is underground or above ground, or it may be rhizosphere, phyllosphere, endosphere. We also exploit insect microbiomes like entomopathogenic viruses, entomopathogenic bacteria, fungi, nematodes. So likewise, there is a human microbiome where we have the gut microbiome, skin microbiome, eye microbiome, or oral microbiome or vaginal microbiome. So likewise, now the microbiome research across all walks of agriculture, health and environment is getting popular. So therefore, how best we can exploit these microbiomes in our day-to-day agriculture, and also how to exploit these things. So the microbiomes, especially when you talk about the plant, they are all progressing. So it all depends upon the age and developmental activities of the plant. So when the plant is in the seed, in the seed germination stage, a community of microbes attracts toward the rhizosphere based on the exudates released by that. So likewise, when it's the developmental stage, the exudates configuration also changes. It also attracts a different communities of the microbes. So here we have the good, bad and neutral microbes. We are only talking about the good microbes which are essential for the plant growth development, and also which are we call it as base microbiomes and core microbiomes. I do not want to go details of these things, but now how to exploit this microbiome science to develop a delivery system. So there is a saying that plants recruit their own microbes, and the plant association with the microbes really gives boost to the plant, and there is a clear intimacy between the plants and microbes. It may be endophytes or maybe the rhizophytes or phylophytes, but there is an intimate association. And at the same time, these associations, how best we can decipher, how to decode these interactions. There is a science and there is a platform now, and how to exploit these things through microbiome engineering. So this says that the tool or toolbox or soft tool, once I met Professor M.S. Swaminathan maybe 23 years back when I entered into the business, maybe 20 years back, he mentioned the microbes are the soft microbial software, something like that at the time. He mentioned, first time I heard, the software is there, but it is a microbial software. He termed microbes as microbial software. So here, today we have the toolbox available. This toolbox, using it, we can understand each and every function of the microbes at a genetical level or molecular level. So we have genomics, enomics, phenomics, metabolomics, transcriptomics, and metagenomics. These are the various tools and techniques available to understand the plant microbial interactions. So this is what actually a small schematic representation. You have gene pool or microbes. These microbes are isolated from agro ecosystems, or maybe from marine ecosystems or desert ecosystems. Where you can have the unique microbial populations are available, bring them to the lab, isolate, characterize, study, and then make use and how to convert them into a delivery system. The delivery system here we call in our agriculture, it may be a granular form, a liquid form, or a powder powder form or capsule or injection. So understand the functional properties of the microbes and also understand the problems of the plants. Now Dr. Venkateshwar, he said just now about soil aggregation. The soil aggregation is because of exopolysaccharide secretes by a set of microbes that helps in aggregation of soil particles. Therefore, the soil aggregates can hold the moisture. So that is one. Likewise, saline tolerance, drought tolerance, and insect resistance, there is a lot of mechanisms exhibited by these microbes can be exploited thoroughly. But only thing is the consortium approach, understanding coexistence of these microbes. Once these microbes are delivered into the rhizosphere, what happens to them? There is a competition from the native microbes. So this is a challenging task for the researchers, but today the science evolving, techniques are available. Definitely we can exploit thoroughly the naturally occurring microbes. So these are the various strategies, and these strategies where you can use, as I mentioned about, you can directly transfer and also you can inoculate and also seed priming. There are many, madam, you can ring at 8th minute. Okay. Okay, so this is how actually the schematic representation, how a microbiome optimization can revive the plant, which is under shock or maybe under disease pressure or insect pressure or sometimes, or maybe the environmental pressure. So definitely we have seen it, we are working it, and there are some companies are coming up. Not only that, there are several, the recent advances we have seen, and also we can see synthetic biology in place. And also there are what we call genetic integrations, gene transformations. So 2 minutes more. So now this is what exactly now the status is. We are now talking about only soil and plant food, but also we have to understand our what we call environment and human health. So One Health people says the concept, it is now, I say that, somebody said otherwise, so one soil, one health. So if you take care of the soil, and we can take care of our health. So there is a serious dysbiosis and also AMR, antimicrobial resistance. It is growing like anything, and therefore we need to strike the balance between the microbial communities, between the three ecological systems. And microbe soil, as I mentioned already, and these are actually the researchable areas and the outcomes. I have listed it, and this presentation can be circulated to all. And this is the theory, that means microbe-assisted agriculture. I know a lot of people are interested in the microbiome technologies, or they are producing either biopesticides or biofertilizers, biostimulants. Now the time has come to understand holistically how to use these microbes into our agriculture. So thank you so much. Hope I have completed. And I will take another 7 minutes for Q&A. Please.
Yeah, Akash, can you send the mic?
Yeah, this one mic.
Yeah.
So I need not to sum up because they have very properly illustrated all the developments with regard to biofertilizers.
Dr. Vimla,
Dr. Vimla Prakash.
you mentioned the change in the FCO regulations for the different. Yeah, I'm Dr. K. Anpurna, former head microbiology division from IARI, New Delhi. I wanted to know, Vimla, what exactly you had in mind when you said that there should be some changes to be brought out in the specifications of the standards for each of these biofertilizers. And I distinctly remember you said something on pH and temperature.
No, that is in case of pH, ma'am, that is exclusively for the liquid formulation. When we are formulating a product as a liquid formulation, the biofertilizer strains, there is in case of liquid, pH is fixed as 6.6 to 7.5, or it is in case of PSB, it is 5 to 7.5. What we are, as an industry, the pain point what we are facing is that I come out with a formulation, I have a very good shelf life, we are having about 2 years of shelf life, but the pH range, it's coming for the formulation is beyond 7.5, it might be 7.8, or it is falling below 6 or 5.5. Since my pH is not fitting into that FCO range, I have to drop that formulation and I have to again rework on another formulating agent. So that is a pain point, because once the any liquid biofertilizer it is taken, it is going to be used as a seed treatment or a soil application. The pH, it's a microbe which is very active in a liquid formulation. The pH is not going to affect after a post application, the pH is not going to do any ill effect on the crop, because in case of chemical fertilizer or a chemical pesticide, the pH has been fixed, because if there is a change in the pH, it shows it's an adulteration or there is any contamination. But in case of microbes, the pH in case of liquid formulation has to be little bit of it has to be a wide
Dr.
range,
Vimla, please
or it should not be.
shorten the answer, please.
Yes, yes. It should be little bit wide. That's a pain point we are facing when we do the formulations. I have to leave that formulation.
No,
That is it.
I understand your point, but I just
Madam,
wanted
kindly
to...
discuss during lunch
Okay.
time, okay? Sorry, you give it to the... Yeah.
Good afternoon, I am Chinmay from knowhow. In the microbiome-based solution, if you are offering, how is the regulatory environment supporting it? Because we are already talking of kind of consortia-based where it's very limited currently, and we are now thinking of a microbiome-based.
See, it is already in place as far as FCO is concerned. So NPK they have already allowed. That is nothing but a consortium. At the same time, CAB RC also actually agreed for, they are now, I don't know exactly the status, but they have in principle agreed for the consortium-based formulations. So microbiome approach, see, so unfortunately the only one issue as of now we are facing is the abiotic stress tolerance. Of course, they want to combine these biostimulants having the nutrition, function that can be clubbed under biofertilizers, but how about the abiotic stress tolerance and also a formulation for that? So still I think we are now dealing with the government to consider it. So as of now we do not have any guidelines for consortium under biostimulants. In fact, I have not at all considered microbes under biostimulants. That's what we are fighting for it.
Yep, true.
Any more questions? Yeah, there.
Yeah, good afternoon to the panel. My name is Komal Bukhanwala from the SML group of companies. My question is to the whole panel. There's a lot of discussion on the mycorrhiza and the beneficial microbes we all know we talked about today. I think one of the presentations pointed out that Aspergillus niger is a beneficial organism for various plant growth activities. While this is BIPA and this is the BioAgri Summit, do you believe that the existing chemical practice that has prevailed has actually destroyed these beneficial microbes over the years, and how is that going to be taken up at CIB long term, right? Because you are trying to put in microbes which the current pesticide practices are trying to destroy.
Venkateshwar. It follow. Anyone? Uday? Appara garu? Yeah. No. Yeah.
Komalji, can you please repeat the question once again, I believe?
So it's a great effort for BIPA and everybody involved with the BioAgri inputs industry to promote organic agriculture. One of the presentations pointed out that Aspergillus is an organism which is important in the soil for plant growth and defense and immunity and all of that. There are pesticides which are being applied rampantly or probably even residing as residue in soil, which are probably destroying those very microbes that you're trying to incorporate in the soil in terms of an input. So how do you bring this to the attention of the CIB or the regulators from a long-term sustainability point of view?
Actually, Aspergillus niger is a well-known phosphorus solubilizer, But commercially, it is not being produced, that much. So therefore, that is not a big.
No, only one product is Kali Chakra from IARI, that is Aspergillus niger.
So it's not very commonly produced, but nevertheless, it is seed coated.
Attacking Aspergillus. So how, when you're saying...
See, there is nothing like a permanent answer for this. It is the science is evolving.
Right.
What you mentioned about the phytoremediation which happens in the soil. So when the set of microbes which are able to survive in the same environment and degrade these what you call pesticide residues, that is one way. So therefore, there is always a competition within the soil between the organisms. So every time you need to identify and characterize those organisms which can survive in that environment.
Okay, thank
So
you.
it's not exactly only Aspergillus.
Yeah, that's
it
one
is for any
example.
organism.
That's one
Yeah.
example. There are many others, yeah.
Dr. Peter.
Yeah. This question can be
This is the last
answered-
question.
Yeah, last question. This question can be answered by either Dr. Vimala Prakash or Dr. KRK. We have studied the efficacy of the biofertilizer when it is coated with the DAP. The stability is also reasonably good. And instead of giving it as a standalone product, so will there be a possibility of integrating with the existing chemical fertilizer in the future? And what is the scope and commercial aspect of this? Because the integration will reduce the burden, like nano urea, the fertilizer added with the biofertilizer. So this is my question.
I think as long as you have proper bioefficacy data tested by ICR institutions or State Agriculture University, plus toxicology data, so you can always apply under FCO or CABRs. So only thing is data you require to develop and prove yourself.